Welcome everyone to Paradise. This is my own lab setting. This is Salima Jan Mohammed with me, and I'm Norman Iscove. And we're kind of interested in science generally, and in hematopoietic stem cells in particular. Our primary focus is on how the hematopoietic stem cells are able to function throughout our entire life. And so that's taken us on a long journey through purification of stem cells and trying to define what the very first stage in the process is between a stem cell that can function lifelong and stages where cells are still multipotent and have many of the features of stem cells, yet these cells are now committed to run forward through differentiation. And in doing so, in recent years, we've defined what I would have to say is a newly recognized layer in the system of cells that can function for months at a time in the mouse and yet not be permanent stem cells. They appear to be cells that are one step forward into differentiation.
What attracted me to the laboratory initially was the caliber of science and the type of research that was being done. So for me, I've been looking at the mechanisms and the genes involved in self-renewal of mouse hematopoietic stem cells. And that's the general theme of the projects in the lab. So, during my undergrad, I had the opportunity to work in a number of different research laboratories, and I discovered that this was something I really got a kick out of. The very first experiment I did was looking to see whether a specific polycomb group gene that we were generally interested in at the time, RAY28, whether it was specifically expressed in hematopoietic stem cells versus the rest of the differentiation hierarchy. And so I had set up my PCR and had cDNAs that we had been previously generated in the lab for every type of progenitor and mature hematopoietic and immune cell. And I found that this gene was only expressed in the most primitive type of HSC and not in even that step forward in that HSC that had lost long-term reconstitution potential. And it wasn't expressed further down at all either. So for me, that was the real moment that captivated me, that this is...
I should have published it. I guess I'm driven by curiosity. I find it an inspiration to understand an element of science. You set a goal and a few years go by and you're closer to it. You did work that explains something, and I think that you want to bring work out that enables in a significant way your field. And I believe that down the road, cure of cancer will actually depend on the kind of information we're going after today.